So as you mentioned, I am a faculty investigator at the HudsonAlpha Institute for Biotechnology, which the main building on our campus is shown on the background there, as well as some contact information. So feel free to reach out with any questions following the presentation, or if there's anything we can be helpful with, depending on your interest. So the usual disclaimer required by AACR regarding that this is a promotional activity and not approved for CME, and the opinions are mine and are not of AACR, which probably no surprise. So our laboratory does a wide variety of genomic research. That gives you an idea of the volume of sequencing work and different projects that have gone up over the years. And you can see as sequencing technology has gotten more affordable as far as depth of sequencing, we've continued to increase the amount of both samples as well as let's see if it works if I stand more over here. And so I'll give some examples of how we've been implementing that over the next couple of slides. But the bottom line is, as sequencing costs have gone down, we've started to introduce higher and higher depth of sequencing, both for RNA sequencing, exome, whole genome, and taking advantage of those costs. Just from a housekeeping perspective, over the last year, we've migrated all sequencing platforms from a diversity of Illumina systems, being the HiSeq X, HiSeq 2500, et cetera, and now normalize them on the NovaSeq platform. So all the data that I'll discuss is from the NovaSeq, and just from the perspective of the final formats is the NovaSeq has a very standardized cartridge format. It's kind of like the Keurig of sequencers. So it makes it quite convenient to use. And then we balance that with both an iSeq platform that we use for quality control, I'll mention that in a few moments, as well as the smaller platforms, the MiSeq and NextSeq. And so again, all of the coming sequencing data I'll mention has been sequenced on one or more of those platforms. So our laboratory works on a variety of projects, everything from metagenomics, forensics, neurological diseases, as well as significant interest in cancer, both pediatric and adult. And then some interesting work in the genomics and genetics of athletic performance and recovery, but I'll focus the rest of the talk primarily on the cancer work. So using a really simplistic Circos view from Cosmic, we can see that there's a huge number of mutations across the genome that have been associated with cancer, lending the idea that both cancer is a complex disorder, as well as importantly, that there are, being able to look genome-wide, both either at an RNA level or at a DNA level, is actually quite important. And then just a quick reminder that although there's a growing momentum in the use of genetic testing to ascribe precancerous risk, BRCA1 and 2 sequencing, et cetera, really only about 10% of cancers have a germline mutation that's attributed to that risk, but certainly the vast majority of cancers have somatic mutations that do not appear in germline tissue. So the somatic mutation rates are actually quite important and certainly quite enlightening when we look at the biology of cancer overall. And of course, there's a number of cancers where you see both of those effects, both somatic and germline. So some of the projects that I'll highlight through this work is essentially two pediatric-based projects. The Gabriella Miller Kids First program, we're one of the sequencing centers for that, where we do both birth defect as well as pediatric cancer genome sequencing. And then the St. Jude Life effort, which is also known as the Childhood Cancer Survivorship Study. And this is a longitudinal study that is looking at approaching 10,000 genomes now. This slide is slightly out of date. 10,000 genomes of individuals who are affected by childhood cancer, survive to adulthood, and they have an annual visit back to St. Jude. So there's a very long longitudinal clinical data set and now genetic data set on these individuals. And a figure from a recent paper on that study is shown here, which the summary of this is what you're looking at is the size of the circle, and the gene indicated in the circle are genes that on genome sequencing were identified as mutated in the individuals that were affected by childhood cancer. Within each circle, the pie chart you're seeing is the proportion of those that had a secondary cancer event as an adult, and then the type of cancer that was present. And so what is being inferred in this is that depending on what the diagnosis of the childhood cancer, followed by deep genetic profiling or genomic profiling, you can make predictions now and do genetic counseling for those conditions. So you can see if you look at RB1, that sarcoma is a very common secondary metastasis. Not surprising, BRCA2, breast, is the primary adult event, and then in a variety of others. And so this kind of validates the idea that for some fundamental cancer positions that you can have a really important downstream overall health monitoring and diagnostic or prognostic monitoring for the rest of their lives. So with this information, we find this as being extremely high utility in the pediatric cancer studies. But as mentioned earlier, only about 10% or so of those with cancer have those underlying genetic mutations. So really a key result, but again, affecting a minority of cancers. And we've been privileged to work with the Oncology Research Information Exchange Network, or Orion, which is a network of now 19 cancer centers managed by a company that was started out of Moffitt Cancer Center called Emtigen. And this is essentially is a primarily adult-based cancer project that unifies the molecular technologies that are used to analyze a wide diversity of samples. And so if we now take some of the summaries of what I mentioned, that now the majority of cancers have somatic mutations, and that pediatric cancers are reasonably well characterized at the germline level to have an impact in downstream, or I should say in lifelong perspectives. On the adult side, the ability to profile somatic mutations, the ability to profile transcriptional changes for the impact in those diseases is quite important. And so as we've worked with the Orion group and their member centers, which all of them are shown there as far as their logos, and then a little bit more information that anyone interested in more information about the program, the website shown at the bottom, orioncancer.org. And so it's very much a new type of research alliance, and really an unbelievably not only interesting, but I think impactful program in that this is as I mentioned, the underlying genetic or genomic technologies that are used are unified across all of the samples. And so an early cut of those samples, both in terms of locations as well as types, are shown here. And so this is 18 months old or so. So you can see, get an idea of both numbers and types of tissues on an earlier pass, where not surprising, the matched normals are mostly from peripheral blood, which is why those numbers are high. And then you can see just an ordered list of the different types of tumor samples that were shown in that. And a large percentage of these were from fixed formalin, paraffin embedded samples, as well as fresh frozen. So there's a mixture through there, and obviously by the title of the talk, we'll get more into the details of the FFPE. But I wanted to give a perspective of the diversity of samples that are going through this process. As far as the platforms for analysis, this is the RNA sequencing technologies are an RNA access based, which is an Illumina product, on all tumor samples with 100 million paired reads per sample, so quite deep sequencing. On the exome, a germline is 20 gigabase per sample, which is basically one S4 flow cell per 96 samples, and the tumors are double that. So it works out to be a 150, 200x germline and a 300x, 400x tumor coverage across the exome. Separate from that project, we've also been quite interested in panels such as TrueSight Tumor or TrueSight Oncology, the 170 and 500 panels, as well as we're working on a custom RNA and DNA platform. But overall, the application of these technologies and the application of these platforms is predicated on, or the resolution within them is dependent on the quality of the samples and the quality of the nucleic acid that we're able to isolate. Particularly if you're interested in looking at whole genome sequencing, where the coverage across the genome will be quite important. And in that case, we have some pilot programs in FFPE, circulating tumor DNA, and even some single cell work. I'll mention some of the efforts in the whole genome in a moment, but certainly some of the coverage limitations that you can get out of FFPE samples are again well recognized, but I think there are some opportunities to continue to push that forward. And then finally, early stage long read sequencing efforts on some of the samples that I'll discuss, and the value of long read sequencing and really helping to understand the genomic architecture, right? The indel and the insertion deletion events that happen are probably as important, and even some early studies are suggesting perhaps more important than the individual nucleotide variation. So the order that these are shown on the slide are going from order of most mature to least mature as far as a technology platform, and and I won't mention any more on the long read sequencing in this talk, other than to say that, again, the quality of the overall material, of course, is important and enabling for this kind of work as well. So overall considerations for FFPE tissue processing, and these are, rather than go through all of the biological considerations, negatives, downsides, et cetera, I'm sure everyone's quite familiar with some of the challenges around FFPE. the level of degradation that's often observed, the base modifications and other damage. And so these are just some general comments and summary bullet points that we've learned over the years across a wide variety of projects. And it notes that tissues are optimally fixed within an hour of recovery, of surgical recovery, and that the optimal fixation time for being able to do molecular studies downstream is between 12 and 24 hours, depending on the tissue type. And that the formalin or the formaldehyde that is used really needs to be a neutral pH to avoid any significant degradation. Fixed tissue should be completely dried before embedding. This is actually a fairly critical part of the process. And then the storage conditions, rather than age, are actually the largest determinant in what the final condition of that tissue will be. So storage of blocks without cut faces prevents essentially unnecessary oxidative exposure. And then the ability to section into a block to get a kind of basically a fresh cut is probably the most important determinant in terms of quality of nucleic acid that you observe downstream. And so with that in mind, tissue thickness for isolation should be between 10 and 20 micron, with a surface area between 50 and 300 millimeters squared. And then it's beneficial if excess paraffin is removed. And that's particularly true in the Covaris technology I'll talk about in a few moments with their AFA methodology. And I'll mention it now for the simple reason is if there's a lot of extra wax around, the acoustic energy essentially will make an emulsification. And so avoiding that emulsification is actually quite helpful. As far as the FFPE damage and modifications, again, I'm sure most of you are familiar, so I'll go through them very, very briefly, that it's often nixed, some double-strand breaks, thymine dimers, hydrolysis of the N-glycosyl bond, and then as well as depurination events, which vastly exceed depyrimidation events, and these are all higher at lower pHs, which again gets to the idea of how the samples are stored, how they're processed, et cetera. It can be quite important. There's both oxidation events shown here and then deamination events. And the deamination events are the ones that are most often seen in the sequencing data as sequencing error. When you have a cytosine that's deaminated to uracil, and then that uracil then becomes a C to T transition in the data. But importantly, that happens on one strand. So it's only on one strand of the DNA, not both. So then what opportunity may exist so you have kind of two opportunities. One is make the sample better or correct. There are protocols. QIAGEN has a product that does a correction of those deamination events, essentially using uracil DNA glycosylase followed by a DNA polymerase to kind of fix that, do that excision repair. There's, I think, a reasonably good protocol for that. Secondly, there's also the idea of taking advantage of longer read sequencing with short insert libraries to actually intentionally sequence both strands. And this can be particularly enabling if you are looking for high sensitivity and rare somatic variants. And the idea is that you'll cover both reads of the DNA with overlapping reads. And what we found enabling for that is now going from a 150 base pair paired end read out to a 250 base paired end read is probably just long enough to make that a viable opportunity. So as I mentioned, as the slide says, the DNA damage related false positives are again present on a single strand. So by sequencing both and then correcting for the common C to T transitions, and then the quite a bit less common other transitions that can happen. So we've tested that or piloted that both on the iSeq, which is a good quality control instrument where you can kind of assess library quality, which is shown here, and you see all the way out to the 250 base reads, which is shown on the left-hand side of the slide. You see a very modest drop-off in quality towards the end. If we look at paired end sequencing, again, a little bit of quality drop-off towards the end of the paired end 250 reads. And then we've piloted this on the S' flow cell with Illumina. So the S' flow cells are not quite commercially launched, but they will be soon. And this is what some of that data looks like, where you again see I know it's difficult to read, but it's low 90% of the data is at Q30 with that same kind of little bit of a drop off at the end. But remember, since we're intentionally making the libraries to sequence from either end of the read, the highest quality of one read is overlapping with the lowest quality on the other. So there's still an opportunity to correct. You don't actually have a circumstance that where the quality drops off, that both reads have that lower quality score. So one read is always a bit higher. And so again, in a pilot phase, if we do whole genome sequencing, it looks like this. So we we prepared what would be considered a kind of a moderate insert library. That's although it's labeled long insert, but this is from the perspective of an FFPE sample. So it's a kind of a long insert FFPE sample. I don't mean to make that confusing. This was prepped with Swift 2S Turbo, which has an enzymatic fragmentation. We used our own in-house adapters and then used hi-fi PCR conditions. So essentially it's a blend of Swift, some of our adapter and primer conditions, and then a Roche PCR enzyme. So it's a bit of a hybrid kit. And did a paired tumor normal sample and sequenced that on an S' flow cell. And so this is 2 samples on a single flow cell. And what we were able to do, and then we also had a control sample that was sequenced separately. And on putting the 2 samples on the flow cell, biasing the coverage higher to the tumor and lower to the normal, you have an opportunity now that on using matched paired tumor normal sequencing on a single flow cell for rapid turnaround time, and you can get coverages in the ballpark of 60x in the tumor and about 30x in the normal. I think ongoing now we're bringing down the coverage in the normal and bringing up the coverage in the tumor slightly. And notice that the insert sizes are in the low 300s, so that paired 250 doesn't quite overlap on every read. If we go to a 250 base pair or less, they actually overlap a little bit too much, because remember there's a distribution of insert sizes, and this is probably about the right spot. These coverage numbers are also exclusive of the overlapping reads, because the QC pipeline that was used kind of throws out the duplicate data. So the coverages are actually close to, if you do actual base coverage, it's about twice that, meaning that, so this is the coverage, if this is the coverage on each strand of the DNA. So the FFPE samples, I think, looked quite good. The overall coverages were very reasonable, particularly in the tumor, because it was sequenced at a higher depth. And again, the sequencing quality was reasonably high. And this is really what you see here. And so this was on those samples on the S' flow cell. So even on the slightly lower quality samples, the coverage looks really quite good. So for an overall workflow process, the samples that went into this. We've also tested it with a more traditional library preparation. So I mentioned the Swift protocol, which was an enzymatic fragmentation. We've done a similar thing in this case here, where we took 600 nanograms to 1.5 micrograms through Covaris standard shearing, so not using enzymatic fragmentation. We wanted to assess if there was a bias introduced by the enzymatic fragmentation versus standard shearing. The answer is there wasn't anything we could detect as a bias in those differences. And then also wanted to see how much of an effect the library preparation method, so we compared it to Kappa hyperprep for the end repair and AD-A. We kept the adapter conditions were identical, so it was our adapters again and primers, and these were dual unique indexed adapters, and then a few cycles of PCR, and then we continued to use the iSeq as a QC methodology for the library. So before going to the large scale production sequencers, they're tested on the iSeq. And then we use Beckman Robotics to actually do the library pooling and normalization based on the data from the iSeq. And so instead of using, for those of you who do a lot of sequencing, instead of using qPCR as a quantitation method, we're actually doing a pico-green based initial normalization and then actually sequencing the library and using the depth of or the performance we see on the sequencer as a way to normalize the libraries. So we're just replacing the qPCR step with a sequencing step. And if we have 96 samples or more in a pool, it's about cost neutral to the qPCR. It's about $4 to $6 a sample, depending on how you do your costing. So with that in mind, we've from a cancer perspective, we've really started looking at the optimization of that full workflow for the analysis, from sample isolation through library preparation to sequencing conditions and then downstream analysis. And certainly that leads you very, very quickly to ask the question, if you improve the quality of the input samples, how enabling is that for the downstream analysis? And that's the punchline for the last few slides. And I think you'll see a fairly dramatic impact. So this is the workflow for the Covaris True Extract kits. It's a dual RNA DNA isolation workflow, and you can see where the splits are. So essentially, it is a AFA enhanced paraffin emulsification. That basically does a gentle lysis of the tissue, gets rid of the paraffin, and then there's the purification and treatment. So that takes about 5 minutes, and then there's about an additional 45-minute step where you essentially centrifuge down the DNA and the tissue pellet, what's remaining, And then the supernatant that contains the RNA goes through one workflow, and that pellet that contains the DNA goes through a separate workflow. So in that case, they happen in parallel. I think operationally in the laboratory, we'll do the RNA analysis or the RNA isolations first. So it goes through this first step and then down through the RNA work. And then all the DNA pellets are held to the side and processed the next day. And that allows a processing of up to 96 samples over about 2 days as far as an overall workflow goes. So there is an additional, a few additional steps on the DNA side that you see on the right-hand side of the slide. And this particular flow chart is taken directly from the Covaris protocol. So for more kind of details on how that works and how it looks, you can, I'd refer you to that. But at the end of that, you have an RNA purification from one arm and then the DNA purification on the other arm. And to automate the process, the thermal kingfisher is, it can be automated on a number of robotics, but we found that to be a really well-matched robotic system where the liquid handling capabilities, cost, and overall throughput works very, very well for the bead-based protocol from Covaris. So marrying the Covaris AFA protocol to the Kingfisher, again, has a throughput of 96, and if you have 3 or 4 technicians working on it, up to 192 samples a day or every 2 days. We've got quite a bit of data on that, and that we've extracted a little more than 22,000 DNA and RNA samples a little more than a year, and they break down into these types for both DNA as well as RNA. So there is a mixture of fresh, frozen, and FFPE samples. They go through the same process, even if they're not FFPE, meaning they still go through this AFA-based extraction technology. But the greatest impact that I'll show you towards the end is on that. Now, I'll mention some of the standard DNA and RNA sample requirements for the different sequencing platforms in terms of just the amount of inputs that are used, and just from a informational or housekeeping perspective. I wouldn't say that these are critical numbers for success or anything else. This is just an idea of, from this isolated material, how much is then used in the downstream applications, and that's what's shown in that page. And I'd be happy to share these slides with anyone who's interested in them. What's more important, and actually critically important, is this slide, that the sample requirements for the extractions, how much goes into this process, is actually quite critical for the success. And that these are the typical yields that are observed. And then the FFPE, where the excess paraffin is removed, is shown in the middle of the slide. 3 sections, about 10 micron thick. If you're doing slide scrapings from about 5 slides, or cores, approximately 3 cores, 1.5 millimeters in diameter and 5 millimeters long. All those are ideal amounts. Now, small deviations on one side or the other are fine, but again, as far as developing tissue specifications, these are actually quite important. So one initial evaluation on one of the very, very first runs, we were looking at the distribution, the DV200 value, which what that is, simply, for those of you not familiar with it, is the percentage of RNA molecules that are at 200 nucleotides or larger in the RNA sample. Meaning, and that the DV200 number is often used for as a benchmark for a number of RNA sequencing kits, particularly from Illumina. There's also you'll often hear reference of a DV300 number that some protocols will use, but again, these are all DV200 numbers, and many of the metrics are at 20% or higher. You can generate a reasonable library, but the higher the percentage, the better. It avoids 5' to 3' bias. It also helps allow more robust detection of fusion genes, and again, just overall ability to work with increase the diversity of the library. The entire process is certainly not automated. And so these next couple pictures are just how some of the benches are set up. The most labor-intensive part of the process is getting the sample of tissue into the Covaris tube. And so these are the larger size tubes that you see on the left-hand side of the slide. And then you can see some of the barcoded, the pre-barcoded tubes that work into that process and some of the downstream settings. And so there is a manual component, and the way the laboratory is set up is there's some specialized heat blocks to hold those tubes, and then there's the use of a large number of expandable pipetters and multi-channel pipetters just to make the process quite reproducible. And the impact of this is pretty substantial. And so what you're looking at, in this case, this is FFPE sections. The left-hand side of the slide is tissues using a standard protocol that was used initially. So this, and we've seen similar results from QIAGEN's AllPrep, Beckman's Formapure, and a few other protocols. And they were all in the same ballpark, that around 20% to 25% of the samples would have a passing metric of a DV200 of greater than 20%. When we had many of the same tissues, but in other cases, some other tissues that had very similar tissue amounts, FFPE block ages, and tissues of origin. And you can see the optimized protocol, which has the foundation of the Covaris methodology shown on the right-hand side of the slide. A full 89 of the 90 samples now had a DV200 above 20%, and you'll notice that a large percentage of samples are exceeding 40%, and then a small number are between 70 and 80% DV200 numbers. This has been very reproducible. Now it's not just 100 samples here, 100 samples there, it's now approaching into the thousands of samples. And so a very enabling ability to now have the majority of samples entering downstream analysis rather than the minority. We actually see a larger impact in cores than in FFPE sections as shown here. So only 6 out of the 79 samples had a passing metric, and with the optimized protocol, it's a full 90 out of 90 with a very, very high percentage of the samples above 40%, and even a larger number above 50 or 60% of the DV200s. So this is particularly enabling on the RNA side, obviously with the molecular weight of the RNA that's coming through, and then as we look at downstream processes and the ability of some panels, which is shown on the bottom of this panel. So this is a screenshot from Illumina's protocol illustrating the number of genes that are targeted in their TST-170 assay, specifically for looking at RNA fusions. And so the longer the RNA, the more accurately you can detect that fusion and the more efficiently that RNA works in their overall process. So you can see there's obviously a growing number of genes that are in that. And so we wanted to answer the question, what is the overall success rate through a panel-based assay like this that has a DNA and RNA component? And that's what's shown here. And so in a separate project, we had 240 FFPE blocks that were submitted for DNA and RNA extraction, and 202 of them had a DV200 value of greater than 20%. And so this is, I think, an important other kind of real-world example that for some cohorts of tissues, there may be a higher or lower success rate than other cohorts. And so we're kind of saying we see an 85 to 90% success rate of these samples, and that is a combination of both the sample pass rate as well as the library pass rate for the RNA processing. So kind of looking at it holistically across the board. In this case, it is an automated processing pipeline. We do 32 samples of RNA and DNA simultaneously on a Beckman I5 span 8, and we've been looking at bias in those as we look at different qualities of samples. And I think the robotic, we've still got a little ways to go of optimization on the robotics. Currently, we can actually see a little bit higher quality of data and a little bit better sensitivity in a manual version of the protocol than the robotics version. But we're hoping to continue to optimize that. But it is a very convenient workflow, but I would say it's not quite the highest performing workflow yet, but it's coming along. Those samples get sequenced simultaneously. What you're looking at is the DNA on one side, RNA on the other, and so this is a 16 samples sequence simultaneously on a next-seq run. So that we've done this to mock a rapid turnaround kind of next-seq workflow if you had a small number of tumor normal samples or a small number of samples going through on a panel where you wouldn't have the luxury of batching large numbers of samples together. So if this was a more clinically based workflow or a translational based workflow, it works pretty well. And one of the things we've seen with the improvements in the overall nucleic acid qualities, the ability to pool the libraries accurately and the relative performance of those libraries has actually been quite normalized. And so it's been impactful just in terms of ease of use and overall optimizations of the downstream protocols as well. Now you'll notice I didn't mention a lot about the DNA workflows on the DNA side. What we found is the impact and the improvement in quality is primarily related to the RNA. There's a modest improvement in DNA, but it's something that I would say I can't illustrate the same impact there. The DNA is still quite fragmented. Part of that, there may be some additional optimizations on that second arm of the workflow in terms of how the rest of the tissue is disrupted, because that probably is one of the keys to this protocol, is how gently you can disrupt the tissue to get the RNA out, and then subsequently how gently you can disrupt the tissue to get the DNA out. Will have an impact on how fragmented that DNA sample is. So to be clear, substantial improvement in RNA quality, an enabling improvement in RNA quality, and I would say a modest improvement of DNA quality, but the yields are still quite good. And as far as the DNA RNA workflow, really quite enabling. So as far as the overall results, So the RNA samples with DV200 scores of greater than 20% increased from 15 to 20% to around 85 to 90%, with many over 90%. We see a higher impact in curls, a higher impact in cores than curls. And we actually see for slide scrapings, the performance is quite similar to cores. It's also quite good. And that's probably more to do with the slide scrapings really avoid almost any of the paraffin coming in. And if you can avoid the paraffin, you see a little bit better performance. So trimming away the paraffin is, again, a quite important spot. So very small cell numbers, spatial type profiling. We've not attempted that. But what you can see, as you get familiar with the Covaris workflow and the different size tubes that they have, I think this isolation protocol and the overall capabilities on that platform will also be enabling there with some appropriate optimizations. As I mentioned, the DNA yields and quality are not quite as pronounced as the RNA, but still, still fine. And what we found is that variable DNA improvement in quality is probably based more on the tissue source and type more than anything else. And again, related to that, how difficult it is to disrupt the tissue and how vigorous that disruption has to be. So finally, in summary, the Covaris, the foundation of this technology, which is from Covaris, being their tubes, their instrument, and the AFA technology, has a clearly demonstrated performance for RNA isolations from FFPE materials. The largest improvement is in the proportion of samples that pass those quality metrics and then go on into downstream preparation. And that we're continuing to evaluate the improvements in the biological resolution of the resulting data. So I think the data I've shown you is very much more on the technical side, right? That you can have a dramatic improvement in the quality of the nucleic acids that come out from a quality control perspective. The next question is how impactful is that biologically, right? What additional resolution of information can you get? So, but I'd say that's an important scientific investigation to undertake. But of course, you can't dismiss the fact that going from 5 to 10% of samples even being eligible for the analysis to 85 or 90% of the samples now being eligible for the analysis, that alone, of course, improves the resolution of the sample. So, but the actual interpretation of those samples, how much, what percentage of genes align to transcripts, et cetera, is an ongoing effort. And we don't want to release that data too quickly and have, kind of any incorrect conclusions. As Jeff mentioned, I'll just mention as well that this entire process, as well as a number of other things, HudsonAlpha has married its abilities in sequencing and bioinformatics with the biospecimen procurement and laboratory processes, et cetera, of Discovery Life Sciences. And so there is a booth here. This agreement was just announced on Friday, so it's early days for this effort, but we're very, very excited about the ability to have truly an end-to-end process and from sample procurement all the way through deep analysis. And so if anyone who's interested in this, or it would be, if the other offer certainly is if you're, because of the the Covaris investment in the protocol and the samples, et cetera, is not insignificant. We're also happy to run pilot samples, et cetera, for anyone wanting to test it before that. And Jeff and colleagues at Covaris or anyone at Discovery can kind of help coordinate that. And then I'll just recognize the research team that did the majority of this work shown on the slide, and I'll be happy to take any questions while I leave that slide up.